So the whole idea with whether or not something is too bright, too loud, too bitter, too sweet, too smelly is all about frequency of action potentials. So our action potentials that we saw before, what we want to know is how often do they occur? If we increase the number of action potentials we get over the same fixed period of time, so say in 1 second you get 10 action potentials versus in 1 second you get 100 action potentials. 10 action potentials will tell you that something is glowing. If we talk about light, this is just relative. These are not actual numbers. But maybe 100 action potentials tells you that something is too bright and you actually squint your eyes. Okay? It is the number of action potentials per time period, not the strength. Nothing changes. An action potential always looks like an action potential. The only time we will see a difference in that is if we go into something called a tetanus state associated with your muscles, and that's a different story. So this actual propagation of nervous signal and information happens in what we call nervous synapses. So this is where signals are transmitted between two neurons or between a neuron and what we call an effector cell. So say you're sending a signal to move your finger, the neuron sends a signal, but the muscle is actually the effector cell. So we've got to get to the point where we can get nerves talking to muscles and so on. This happens initially by electrical synapses, action potentials as we were talking about, but the problem is when you get to the end of a nerve, we get to something called a synaptic cleft, or a space between two nerves. And we've got to jump that space. We do it chemically by something known as a neurotransmitter. The neurotransmitter is going to be released by the presynaptic cell and picked up by the postsynaptic cell. So if this is our presynaptic cell and our postsynaptic cell is at the bottom, remember nervous signal only transfers in one direction. So we have it coming in from the top. We get an action potential at the end, but we run out of membrane. We get to the end of this nerve cell. So this is the axon of this nerve cell, and this is the dendrite of the next nerve cell. So remember, it's always dendrite, cell body, axon. So when we get to the end of this axon, we're going to release neurotransmitter that's going to be picked up by protein receptors on the surface of the next nerve cell. And they've got a big picture over here to show you how they would connect together. This synaptic cleft is what we have to get across, and a chemical signal is a much more efficient way to do this than trying to send electricity across an open gap. So what happens is the neurotransmitter is actually going to bind to this receptor and allow ions to come in. What kind of ions do you think these are? What ion channels did we open when you got a signal to start an action potential? Sodium ions. So these neurotransmitters are chemical signals that are going to come in, act on this enzyme, this facilitated diffusion channel. It opens the channel and it allows sodium to come into the cell, starting the next action potential. When the neurotransmitter gets broken down, the ion channel closes, sodium stops, and we go back to rest. Neurotransmitters come in two forms. They're either excitatory or inhibitory. So the one I just described to you with sodium channels is what we would call excitatory. It means that it's going to cause the next cell to actually send an action potential. Inhibitory decreases the ability of a cell to send an action potential. So if you're decreasing action potentials, you're decreasing the signal transduction. If you're decreasing the signal transduction, the receiving neuron cannot fire and you get a stop. You won't conduct any more signal. Now, the problem is the receiving neuron that we mentioned, the postsynaptic neuron that we mentioned, after the gap, right? It can get both excitatory and inhibitory signals. So what it does is it takes this information and makes a decision. So it's going to weigh the excitatory and the inhibitory, and then there will be a stimulus only if the excitatory is greater than the inhibitory. Then you're going to get an action potential or a stimulus to keep sending that signal. So this is what we start looking at as summation. The nerve has to figure out what it's going to do. Is it getting enough excitatory stimulus to continue? If not, if inhibitory is greater, then the nerve stops sending signal. When we put a bunch of these nerves together, we start seeing how a more complex response is possible. So here showing you just excitatory and inhibitory signals coming in. These would all, the yellow here are dendrites, but the white are axons from the presynaptic cell. And you see they're coming in from all over the place. So we're often funneling information from a lot or a very large area into a signal pathway that is much more condensed and concise as we go. We've got a couple of different types of neurotransmitters. Acetylcholine is the one we see probably the most, associated with both your brain and the peripheral nervous system, and it's critical for reflexes and muscle movement and so on. We see biogenic amines that are part of the central nervous system. Norepinephrine is probably another one you've also heard of, associated with the central nervous system. This would be important in your fight or flight response. You've probably heard of this before. Are you going to run away from the danger, or are you going to face it and fight it? Very different responses for your body to do that. Neuropeptides are involved in something known as endorphins, which you may have all heard of already as well. And this is actually, endorphins determine how you perceive pain. And then nitric oxide is actually one that we see most often associated with erections in men during sexual arousal. So very different types of neurotransmitters, right? Depending on what their job is and where they're found in your body. Everything we do can affect how neurotransmitters behave, particularly things that we put into our system. So if we look at things like psychoactive drugs, so if you're talking about things like morphine or cocaine or PCP, anything like that, these psychoactive drugs will actually impact the synapses and keep neurotransmitters from functioning properly. Caffeine actually counters inhibitory transmitters. So when we talk about caffeine, we talk about it as being a stimulant, right? It's keeping the inhibitors from acting properly. Nicotine, also a stimulant, does the same thing. Alcohol, however, depressant, right? And this will actually increase the inhibitory neurotransmitters. So if you take these things together, you have to keep in mind what's going to happen with your system. So when we look at folks that smoke nicotine as a stimulant and drink alcohol as a depressant, what's going to happen to your system? Or a very popular drink anymore is Red Bull and vodka. Red Bull's chock full of caffeine, right? Vodka's alcohol. It's a depressant. Stimulant and depressant countering one another and fighting against one another, and your system trying to deal with that balance.